All right, so thank you everyone. Good morning and good afternoon for those of you on the East Coast. My name is Tina Delas Reyes, and today I'll be talking about advancing mRNA therapeutics with Oxford Nanopore Technologies as a primer before the Eclipse Bio webinar. And in recent years, there's been growing regulatory recognition to incorporate sequencing-based assays into QC. This has come down from various agencies discussing the incorporation of full plasmid QC and vector sequencing in through mRNA sequencing for both identity and integrity, as well as looking at viral contaminants with next generation sequencing. And in order to capture this, Oxford Nanopore is meeting this moment with end-to-end solutions and the development of a 21 CFR Part 11 platform. And with this compare and contrast, you can see that we're consolidating all of these legacy QC pipelines into one single direct sequencing assay. Previously, things would require complex sample prep and possibly even cDNA conversion for the likes of mRNA vaccine QC, whereas on the Oxford Nanopore approach, we have a unified multi-attribute assay that looks at identity, integrity, and poly-A tail length. Legacy approaches may contain high data analysis overhead as you incorporate multiple attributes into one, whereas our automated epitome analysis requires zero coding and provides you with a pass-fail output at the very end. Legacy approaches also have fragmented data generated over several days, increasing your time to answer. Whereas with the Oxford Nanopore approach, you get actionable results generated in hours. And on that last point, I do think it's important to explain how Oxford Nanopore sequencing technology works in order to orient you around that technology and give you a richer insight as to how we're able to capture all of these different attributes. So when I say live base calling, this is because Oxford Nanopore sequences and provides data back to you in real time. Our platform is built around our core nanopore chemistry, made up of a nanopore, a motor enzyme, and a motor protein. And the three of those components bring your molecule directly to the pore. And as your molecule is passing through, whether that be DNA or RNA, it perturbs the electrical current. And that squiggle on that electrical current is actually a technical term in Oxford Nanopore. And that is what we are actually reading out in order to capture your different base readouts. Because of this, we are the only platform that allows us to sequence both DNA and actually RNA directly without using PCR. Direct sequencing of these molecules means that all of the different modifications are also maintained in the data. And this is going to become incredibly critical as we talk about the different modified bases that are important in mRNA therapeutics. So what does all of that nanopore sequencing background mean for you? It means we can consolidate the assay into one workflow, capturing edits, off targets, structural variation, and methylation together. You get richer insights because you're able to actually capture the native DNA and the RNA as opposed to the synthesized strand, complementary strand that it belongs to each of those molecules. You get real-time analysis, meaning instead of loading your platform and waiting 3 days for the whole run to complete, you can see the data being generated as it's happening and then end your run so that you can move on to the next step of your workflow. We're also read length agnostic. We can look at any read length from 20 nucleotides up to 4.3 megabases, although technically there's no upper bound. And platforms, and we cover platforms from discovery in through GMP with both research use only instruments and 21 CFR Part 11 options. As I mentioned, you will get accurate detection of modified bases at the click of a button. So there's no extra bisulfite conversion, no additional prep necessary, which means you save both time, cost, and actually your sample material. You can see here on the right-hand side, all you have to do is click a button to turn modified base calling on. So no matter what you do, those data are being generated. It's whether or not you want to capture it for your own data analysis. And those modified bases become super critical when we're doing things like mRNA vaccine QC. While you see this is talking about mRNA vaccine, this does apply across all mRNA or RNA therapeutics as well, because we are able to capture the direct molecule. So for sequence identity, we are able to look at the sequence confirmation and leverage the modified base caller. Most critically here, we're talking about the use of our N1 methylpseudouridine base caller, which can capture that modification that's become incredibly critical in RNA therapeutics. For integrity, we can look at intact transcripts, and we do have 5' cap currently in development. On the purity end, we have double-stranded RNA in development, but we do have now the capacity to look at your poly-A tail length estimation. Again, all of this happens on one platform, and we run this on our GridION device, which runs MinION flow cells. And Wayne will talk about this later, but we also pair the mRNA test along with our plasmid precursor QC test as well. We believe this represents a new gold standard for mRNA QC by simplifying the workflows, reducing your turnaround time, and providing richer, more accurate insights into mRNA medicines with just one assay. So how do we actually do this? You do get same day results with a library prep protocol that's relatively straightforward. We recommend starting with 300 nanograms of mRNA. And the first step is to add the adapter, and wherein the adapter is annealed to the poly-A tail. This serves as our starting point for a reverse transcription. I mentioned earlier, we are looking at the RNA directly. We do this RT in order to confer some stability, but ultimately, because the nanopore sequencing adapter isn't attached to that cDNA strand and it doesn't attach to the motor protein, we are reading only your molecule of interest, and that cDNA strand does not get sequenced. Base calling happens in real time for this purpose, and we use both the canonical bases as well as the modified bases, specifically N1 methylpseudouridine, in this purpose. After the base calling is completed, you can feed the sequence through our data analysis pipelines for the QC analysis, which are available in our Q-Line platform, or you're welcome to build your own tools in order to capture these data. So in summary, the direct RNA sequencing reveals even more with high raw read accuracy at 98.9%. We do have a high output where we typically in nanopore land think about the nucleotides number of bases generated because we are read length agnostic. But since we're talking about RNA, we look at 25 million reads for longer fragments or even 100 million reads for shorter fragments that are greater than 50 nucleotides. We have multiple modifications that can be base called. So while I've called out N1 methylpseudouridine earlier in this presentation, you can see here that we have M6A, M5C, pseudouridine, et cetera, all included in our Minnow and Dorado base calling units. We have a long read N50 of 1.8 kilobases and a short read sequencing where on the particular platform of direct RNA, we capture greater than 50 nucleotides. And I do want to take a moment to talk about the accuracy, as I often get this question when speaking with customers about Oxford Nanopore. As you can see here under in the third bar, the Medaka consensus Q score per base. I like to call out that when we talk about Oxford Nanopore Q scores, we're often referring to a single molecule raw read accuracy, which is what I stated on the earlier slide of 98.9%. However, when we actually capture the consensus and look at your stacked reads up against the alignment that you're looking for, we actually are able to increase that Q score quite a bit. So what you'll note here is actually Q score of Q50, which should give you all of the confidence that you need to call bases with this high level of accuracy. So we're happy to walk you through this in further depth if you have further questions. But for now, I'd also like to feature other key workflows in biopharmaceutical development. If you work with modalities outside of mRNA, or if you're up or downstream of that development chain, we also have workflows that can meet your needs. We have fit-for-purpose NGS solutions for whole plasmid sequencing, which involves poly-A estimation there as well, and a look at the full plasmid. For adventitious agent testing, we have a long history of pathogen surveillance and microbiology, and we've been able to leverage that to look at contaminants in production batches through either targeted or metagenomic approaches. In antibody development, we have customers using phage or yeast display libraries, doing immune repertoire analysis, replacing their legacy Southern blot assays, and doing cell line characterization with nanopore sequencing. And lastly, on the vector QC and insertion site analysis side, we can characterize cell lines, look at AAV, and particularly LVV due to our direct RNA assay, and then also do whole genome or targeted sequencing methods in order to look at those insertion sites. And one additional note is that Oxford Nanopore has adaptive sampling, which we refer to as virtual panels. In adaptive sampling, you leverage a bed file as opposed to looking at PCR amplicons in order to look at targeted regions of the genome. And you can use that to do insertion site analysis or other targeted sequencing needs. And much like direct RNA QC, this is unique to Oxford Nanopore as we are the only sequencing company that can do this. So, with that high level primer, it's my pleasure to now transition into an introduction for Dr. Wayne Doyle, our head of scientific platforms and strategy, as he talks us through the offerings with Eclipse Bio.
Thank you very much for both Jen and Oxford Nanopore for having me today. I'm excited to walk you all through how we use nanopore sequencing at Eclipse Bio to generate the kind of high resolution data that RNA drug developers need to help make more confident decisions on the therapeutic quality of different RNA drugs. And so before diving into the data and how we specifically apply nanopore sequencing, I did want to take a step back and talk about why RNA matters as a drug modality overall. And so each year, there's over 13 million people die from infectious disease, in many cases because there's not an effective vaccine that's available. In addition, cancer takes another 10 million lives annually, and treatment resistance remains a major hurdle to preventing these deaths. And then for rare and genetic disorders, there are over 300 million patients worldwide. The vast majority of them have no approved therapies. And while traditional small molecule and biologic approaches have brought tremendous progress, they often manage symptoms rather than address the root causes, which can prevent cures and has led to many of these diseases being viewed as undruggable. But RNA-based drugs, where use a long nucleic acid, such as mRNA or circular RNA, offer a fundamentally different approach. Let us go beyond treatment to potentially cure and prevent disease at a molecular level. And we are already seeing this play out. RNA therapeutics span an enormous range, from mRNA vaccines that proved themselves during the pandemic, to gene editing therapies, personalized cancer therapies, and more. And the breadth of what RNA can do as a therapeutic modality is really unprecedented, and it is becoming a fourth pillar of how we approach in modern medicine. But there is a challenge developing these therapies. And because getting an RNA therapeutic to work isn't just about having a good sequence on paper, efficacy depends on getting multiple stages right, starting first with the design itself. You need to choose a protein to express that will have the desired effect. But then you also need to optimize the codons for stability and translation to make sure the protein is being produced, and select UTRs that will limit off-target activity. And then once that design is complete, it then has to be manufactured. So it has to be synthesized at scale with high fidelity, and then efficiently purified and encapsulated into a delivery vehicle. It then needs to be purified efficiently and then encapsulated as well. And then even after that encapsulation, it then needs to be delivered into the body, at which point it has to be taken up by the target cells type, released into the cytoplasm, and then translated effectively into a protein. And each one of these steps has to work in order for it to be effective. And a failure at any single point, a truncated RNA during synthesis, double-stranded RNA impurities, a truncated poly A tail, can derail the entire therapeutic. And that's why quality control matters so much, and why regulators and advisory bodies are becoming increasingly focused on quality control across the long nucleic acid space. As one example that I'm showing here, the US Pharmacopoeia has recently published draft guidance specifically for mRNA vaccines and therapeutics, laying out what critical quality attributes need to be examined during the entire production process, which I'm showing some of those attributes here. Such as what is the identity of the plasmid, what's the identity of the RNA sequence itself, is it potent, is it stable? And for these, there's typically specific analytical procedures that are recommended. But an open question is the field, are these tools that we're using give the resolution that's really needed to drive good decisions, to create very effective therapeutics? And that's because there is a disconnect. RNA-based medicines generally are cutting edge. First approvals, of course, during the pandemic in 2020, first personalized gene therapy in 2025, and the science is moving incredibly fast. But the analytical tools that are used to validate these medicines, many of them are more than decades old. Dot plots, ELISAs, columns, gel electrophoresis. They're workhorses, very well validated, but they also give typically bulk readouts when it can be a benefit to also get molecular level resolution. And so it can be important to then get more detail, which I can just show an example of kind of this bulk level and what can be missed. Looking at a dot blot, which is an approach for measuring double-stranded RNA, a critical impurity that's generated during in vitro transcription. And if it's retained, it can trigger innate immune activation and degradation of the therapeutic. And so with the dot blot, use an antibody against double-stranded RNA, and it gives you a very nice bulk quantification of here's the total amount of double-stranded RNA that's present. What it doesn't tell you is what is the actual double-stranded RNA species that are present? Are they changing from lot to lot? What's producing it? And potentially, are there biases from the antibody that you're missing specific species being present? And this is just one example, but each of the standard assays that I showed earlier has similar limitations. You typically get a number out, but limited mechanistic insights to help fix a problem if one is present. And this becomes a problem depending on where a quality control issue gets detected using these assays. During early stage R&D, if there's a design choice that leads to high double-strand RNA or poor yield, that can be caught relatively easily, although without high detailed information, you typically have to iterate and optimize until you get to an ideal situation, which can increase costs. But if a quality control issue is not missed until later in preclinical development, it could lead a drug developer to miss a prescheduled slot with the CDMO, extending the time for it to actually be produced, leading to months of delays and significant costs. Or in a worst case scenario, a quality control issue isn't caught until an actual clinical trial, which can lead to a halt of that trial or even a termination of the program. Which makes it critical that as the field continues to advance and we continue to expand the types of therapies that are being produced, that quality control also advances as well, so that we can capture issues and identify how to fix them sooner in the process. And that's where sequencing can come in as a complement to the existing approaches. And with sequencing, both direct RNA sequencing, as I'll focus on today, and then other sequencing-based approaches, you can use specialized library preparation protocols to enrich RNA to examine specific quality attributes, such as what's the structure of an RNA, what impurities are present, what regulations occurring. And then once you create a sequencing library, you can then run it through a sequencer, such as Oxford Nanopore's GridION. That allows you then directly look at each nucleotide that's present within the sample. And then a variety of bioinformatics analyses can then reveal the quality of the therapeutics. And the resulting data then provides nucleotide level resolution. So you don't just get information on whether something passes or fails, but you can get into why is it passing, why is it failing, what can be done to actually improve the therapeutic moving forward and make sure that the best candidate goes into the clinic. And this type of sequencing support is what we do at Eclipse Bio. Since 2017, we've been developing sequencing-based approaches to help researchers and drug developers design with precision for their therapeutics and then optimize them and characterize it. We have worked with over 100 partners, including biopharmas and biotechs, to apply these methods across the full life cycle of RNA drug development, with our platform really being built on the principle that better data leads to better decisions and more effective therapies for patients. And if we look at what sequencing can do in detail in relation to standard critical quality attributes for RNA therapeutics, we can actually see that sequencing addresses really the full spectrum. It can be used to look at the identity of an RNA, as I'll show in this presentation. Are there specific bases present that should be there? Also structural identity as well. Is it folding the same way each time? Integrity, does the RNA remain intact? It measures of purity, stability from the drug substance to the drug product to within the cell. Multiple aspects of potency, from is the drug product actually being taken up into the cell? Is it releasing? Are there potential issues with translation? And more unbiased approaches to looking at how is the cell actually responding are the potential safety risks across the entire transcriptome and the entire translatome. And while sequencing can be applied across multiple of these critical quality attributes, for today I'm going to focus specifically how at Eclipse Bio we use Oxford Nanopore sequencing technology to examine key aspects of identity, purity, and stability. And I'm going to focus first on identity and looking at what happens at both the DNA plasmid and the RNA level during the production of an RNA therapeutic. And so just to set the stage a little bit, when we look at synthesizing an RNA therapeutic, be it a gene editing therapy like Cas9 or a vaccine, what's typically done is you start with a plasmid template that encodes your specific therapeutic. That then gets amplified in bacteria and then transcribed in vitro. While this is very effective, at each step there can be potential issues. There can be synthesis errors. You could have truncated full-length constructs. There can also be mutations that occur during recombination during the amplification, or base misincorporation during the in vitro transcription. And so it's important that the quality control approach that's used is one, able to capture these, and then two, get insights into where the specific issues have occurred, or if there's specific sequence designs that are contributing to these quality control issues occurring. And we have found that Oxford Nanopore sequencing gives us exactly that visibility that's needed. At the most fundamental level, it does reveal the identity of each molecule, so you can use it to look at, have new regions been added to the sequence? Have there been deletions that you've lost part of your therapeutic? Or have there been mutations and that bases have been changed? And the way that this works, as Stina has gone through as well, is that either the DNA or the RNA is fed through a pore that generates a distinct electrical signal that then an algorithm can flag and say, this is the specific nucleotide base that's going through. And that can then be used to say, are there unexpected changes in the sequence, either in the plasmid itself or polymerase errors during transcription. So this means that we're not relying on indirect measurements. We're directly reading what is actually occurring on the specific molecule that is going through the pore. And I'm just going to show some examples of this in practice. Starting first with looking at unexpected changes in the sequence. I'm just showing here an example where we're looking along the length of an IVT RNA, and then the y-axis is showing the percentage of bases that match expectation. If you get a decrease, that indicates the wrong base has been incorporated or there's an issue with the plasmid fidelity. I'm showing here for an example with the orange arrow. This is a base that almost 100% of the time, the wrong base is incorporated, indicating that that plasmid had a sourcing issue and the wrong sequence was present. That was looking at the IVT RNA, which I'll go back to in a few slides, but we can actually just look directly at the plasmid as well and capture this even before the synthesis. And so I'm going to show a test case in which we designed a Cas9 therapy and then encoded it within a plasmid. We then transformed 6 colonies with the same plasmid and then evaluated them with nanopore sequencing to look at the quality of each colony. And I'm showing the results of that here, where each of the different colonies is along the x-axis, and then we're looking at the number of deletions that are present, or if there's insertions or base mismatches. As you can see, each of the colonies all had a single deletion present, which if we actually look along the length of the plasmid, all the deletions were at the exact same site. It's specifically at the start of a homopolymeric stretch, a region that's likely to undergo recombination E. coli during amplification. And so this is an indication that with the specific design, this can be an issue. However, this isn't a non-functional part of the plasmid, so it should have no effect on the ability to actually do the synthesis and get a final therapeutic. Where we were more interested, though, was in the deletions that are present in sample E. And so with this data, we don't only quantify the amount of deletions and mismatches that are present. We can actually look at and say, where did those mismatches occur? What is the actual functional impact of having those? And so I'm showing here along the length of the plasmid, and then showing the frequency that we actually see either a mutation occurring or that deletion. So the deletion is the one that we saw earlier. There's no functional impact to this. but what we're more concerned about were the mutated bases. One of them that was present almost 100% of the time across all the DNA sequence that did mutate 3' UTR. So while that could have no functional impact on the exact amino acid that's produced, this could have impacts on the amount of RNA that's produced or the cell type specificity that occurs. What was more concerning for us, though, was the other mutation that was present. This actually led to a valine to become an in-frame start codon. So if this plasmid had gone ahead to synthesis, and it could potentially lead to a different protein product coming out that would be unexpected. And so with this, we can then screen across the plasmids and say that clone E is not one to move forward with. It will be problematic in terms of what proteins are produced. We should instead move forward with A, C, D, or F. This is something that's very quick to do with the nanopore sequencing and provide really high resolution data to it. But this is just one stage of looking at the identity of what the final RNA will be. It's also important to look at what's actually occurring during synthesis as well. And that is something that we can do with nanopore sequencing as well. Looking at, once we use that starting template, what RNA comes out at the end. And so I'm now going to show a different RNA that was produced in which we gave an RNA sequence to two manufacturers. Manufacturer A was a large CDMO with GMP manufacturing capabilities, while manufacturer B was a smaller contract manufacturing organization more focused on research use only. We gave them both the same exact RNA sequence. We asked them to produce it with just standard uridines, so no base modifications present, using their standard plasmid sourcing, their standard manufacturing processes, and then asked, do we see differences in the quality based off sequencing between the two manufacturing runs? And so first we looked at the base identity, which is present here. This is similar to the plot that I showed before, in which along the x-axis is each nucleotide position of the in vitro transcribed RNA. And then the y-axis shows the percentage of times that a base was called correctly. So if it drops below that 100% baseline, that indicates that in some percentage of the time, the wrong base gets added. What we did see is that both manufacturers have very similar amounts of changes in base identity. Majority of the bases are exactly what we expect to see. But there's a G that gets read as an A in about 10% of the RNAs that are profiled. That does lead to a change in protein product, so that is problematic. But that occurs both in manufacturer A and manufacturer B. There's also a C that gets read as a U. That occurs about 20% of the time, but that is a synonymous change, so it leads to no change in the final protein product. Although depending on the tRNA levels that are present within the cell that's being transfected into, that could lead to pausing if it leads to a codon that's not represented enough in the tRNA population. But overall, both manufacturers having similar levels in base identity indicates that their plasmid supply is comparable, their polymerase fidelity is comparable as well. This is more something that we typically see when there's a sequence design component that makes it harder for either the plasmid synthesis to occur, or the polymerase can run into issues and start incorporating bases due to incorrect bases due to the local context. But sequencing was able to allow us to tease that apart by doing that direct comparison between the two manufacturers. This was specifically with unmodified bases, though. What historically has been a challenge with nanopore-based sequencing is looking at modified bases. So this is related to N1-methylpseudouridine, base modification that really enabled the success of the COVID-19 mRNA vaccines. Because the incorporation of this base leads to a drastically reduced activation of the innate immune system. So when RNAs are introduced into a cell, the cells can recognize that as a potential foreign nucleotide sequence, and that can lead to innate immune activation, leading to inflammatory responses, degradation of the therapeutic as well. Incorporation of N1-methylpseudouridine reduces that effect. But this can be a challenge if you don't have an effective base caller, because that leads to a different electrical signal. And so with the standard base calling approach, Often uridines will appear as mutations because with that M1 methylpseudouridine, just looks like a different electrical signal coming through. So something that we're excited about is that Oxford Nanopore, as Tina's mentioned, has launched an updated base calling algorithm that correctly identifies M1 methylpseudouridine bases as uridines rather than mismatches. So I'm showing that here. This is for a M1 methylpseudouridine incorporating RNA. Where on the top, we're showing the canonical base calling, and then at the bottom, showing base calling with the M1 methylpseudou caller. Gray are bases that are called matching the expected sequence, and then purple or red are mismatch calls. And as you can see with the new caller, we have a drastically reduced number of mismatches. We're now only looking at mismatches due to an actual problem with the polymerase incorporation or the plasmid identity, allowing us to then get deeper insights in the quality of these modified incorporating nuclear therapies. And as you can see, we also get higher resolution across the entire length of the transcript as well, including at the end of the transcript with the updated base caller. And so this is a significant step forward, allowing us to accurately assess the identity of modified RNA therapeutics, allowing this analytical approach to be used across the vast majority of the clinical pipeline. And we are excited at Eclipse Bio to start offering this soon as part of our characterization services using sequencing. And so that was an example of looking at a few examples of looking at the identity of therapeutics, both in terms of the plasmid, but then also the RNA, ensuring that when it enters the body, the correct protein will actually be produced. But that is just one slice of the different critical quality attributes that need to be examined. One other dimension is even if the identity is correct, the RNA also needs to remain intact. That's critical for the actual protein production and the long-term stability. But this is a challenge with RNA therapeutics. RNA is inherently labile. It can undergo hydrolysis and oxidation that leads to degradation of the therapeutic, loss of protein production. This can occur through autohydrolysis with the RNA interacting with itself, or through interactions with components of lipid nanoparticles. And we can see the effect of that degradation here on a fragment analyzer trace using capillary gel electrophoresis, where you have the intact RNA. And then this was after heating. We do get fragmented RNA occurring as well. And this can occur due to specific sites that are more prone to undergoing hydrolysis. With traditional approaches, such as CGE that I'm showing here, we just see the fragmentation occurred, but it's hard to see exactly where did the break occur, what specific nucleotides were involved. Is it a consistent break that occurs every single time? With nanopore sequencing, we can start to approach that. So what we can do is we can look across the entire length of the RNA, shown here along the x-axis, and then plot the amount of sequencing data that we get at each nucleotide position. So in a high-quality RNA, like I'm showing here from both manufacturers, we should see a pattern like this, that we're getting very equal coverage along the entire length of the RNA. This means that we're not really getting any types of breaks. If there was a fragmentation occurring, you'd see a drop of coverage because you're not able to actually sequence along that entire length. So in this case, both manufacturers produce high-quality RNA with comparable levels of intactness that's present. But in addition to the integrity aspect, another component that can be examined with nanopore sequencing is also looking at other aspects of purity, including the length of the poly A tail that is present. Which, just to give some background for the poly A tail, each component that goes into the design of an RNA therapeutic plays key roles in its ability to be translated, stable, and have low immunogenicity. So open reading frame, that encodes your protein of interest. It's important for the stability of the therapeutic. If you have unstable bases, like unpaired bases in that region, that can lead to breakage that can be detected with that RNA integrity analysis. The UTRs also play key roles in stability and translation, allowing for cell type specificity. The cap plays significant roles across multiple dimensions. But the tail is also critically important for informing both the stability of the therapeutic and the translation. And this is due to the loopback of the tail being required for the actual initiation of translation and protein production. and the length of the tail preventing degradation through deadenylation processes. And so this has made it critical that for this therapeutic development, that poly A tails are incorporated and at appropriate lengths. And so for this in therapeutic development, there's typically two main approaches. The first is where you encode the poly A tail directly within the template. This then allows you during in vitro transcription to produce it. This is a very simplified workflow. It allows you to get very defined poly A tail lengths. Although there are limitations, there's a maximum length that you can get because E. coli do tend to do recombination on homopolymeric stretches, including poly A tails. So typically around 120 nucleotides is a common maximum length that's used, although adding linker regions can extend that. Alternatively, there is an enzymatic approach in which you do in vitro transcription on your therapeutic and then use an enzyme to add the poly A tail. That's template independent, but since you're using an enzyme, that does get variable tail lengths, although they can be longer, allowing for potentially longer stability. With nanopore sequencing, as it's going through the pore, we can directly look at the length of the tail by reading these adenosines at the end. And we can see the difference between templated tailing and enzymatic tailing. So on the left, I'm showing a distribution of poly A tail lengths on a templated. So it's set to be that there should be 120 nucleotide long tails. And we do see a very tight distribution centered at 120. So the length of the poly A tail is measured across all in vitro transcripts using direct RNA sequencing is here. And then the percentage of them is along the y-axis. On the right side, I'm showing an example where there's enzymatic tailing, in which case we see a much wider distribution consistent with variability in how the enzyme is adding the tail to it. That's two test situations. I did want to go back to the manufacturers and that I discussed earlier, where we can now see a strong quality difference between the two manufacturers. So to date, we had seen that the identity was comparable between them and the integrity was comparable. But we then also looked at the tail, where for manufacturer A, the large CDMO, their tails were centered at the expected 120 nucleotides and narrow distribution. This is what we typically see with high quality RNA. For manufacturer B, though, we did see the tail lengths were shortened and also highly variable. So we did have centered at 120 a small peak, but then we also have a number that are much shorter, even going to no tails that are present. This is something we see very often if there's a lot of recombination occurring in the E. coli during amplification, typically indicating kind of stress on that E. coli health. And so this is something that would be potentially missed with some other approaches, but here we can tease apart. There's a specific change in the quality and the length of the tails. Which we look now across the manufacturers with sequencing approaches that I showed today using nanopore. We can see with manufacturer A, they have the full length, the expected identity, and the expected tail lengths. Manufacturer B also has the expected full-length RNA present, expected base incorporation, although they do have variable tail lengths, that if this was delivered into a cell, that would lead to lower translation, lower stability. That if you're just looking at a final protein output, it would be hard to tease apart. Why is that protein output lower at this manufacturer? But here we can see it's due to the tail length specifically, which then allows us to make more informed decisions about the specific manufacturing process. So that was a few examples of how sequencing can be applied to look at the quality of RNA therapeutics. But sequencing-based analytics can address really across the spectrum of different critical quality attributes. I didn't discuss it today, but with our eCompass platform, we also use sequencing to look at potency, determining are there ribosome pauses, how long is it taking for the lipid nanoparticle to be taken up and have the RNA released into the cell, double-strand RNA assessments as well, what species are present. And at Eclipse Bio, we offer these sequencing capabilities really across the full preclinical development lifecycle, from AI-powered design to candidate selection, process optimization, and then characterization of both the drug substance and the drug product. And we provide this as full preclinical support that you can come to us with an amino acid sequence. We'll help you design it and validate it versus doing targeted support specifically where your program is at. So helping examine your manufacturing process, helping to characterize the final drug product as you move towards regulatory filings, with the goal of providing you the insights that are needed using sequencing to make better decisions and then move forward with confidence into the clinic. With that, I'm happy to end, and thank you very much for listening to my talk.